Hello everyone, I'm Qianwen Wang, currently a postdoc at Harvard Medical School. Today I'm presenting our study, ThreadStates, state-based visual analysis of disease progression. This is a joint work between Harvard Medical School and the Dana-Farber Cancer Institute. This study proposed a visualization method to analyze cohort start data, explore patient status, and understand the possible progression patterns of disease. To start with, what is cohort start data and why we want to record and analyze it? Cohort studies are a form of longitudinal studies that recruit patients who share a defined characteristics and follow those patients over a period of time. Cohort study plays an important role in clinical research. Those studies contribute extensive patient observations across multiple time points and offer valuable information for researchers to check patient status and understand disease progression. For example, in the study of Alzheimer's disease, by recording and analyzing biomarker values across multiple time points, researchers can better understand the possible progression paths and the causal effects of certain drugs. In our previous study, we present OncoThreads, a tool for the visualization of longitudinal clinic data. This tool visualizes patient cohorts as tempo heat maps and syncing diagrams, and supports the interactive exploration of a wide range of clinic features. OncoThreads is also presented at this year's BioVis conference as a proceedings presentation. However, the analysis of cohort study can become increasingly challenging with extensive data sets. With a large number of observations across multiple time points, it's difficult to extract meaningful insights. Therefore, in order to improve the analysis scalability, we propose ThreadStates as an extension of our previous study. We aim to address three scalability-related challenges. The first challenge is related to the number of patients. A cohort study usually includes a large number of patients. While those patients share a certain defined characteristics, such as they have the same disease, but they may also have different genders, ages, and other patient-level features. Second, each patient may have different lengths of observations, making it difficult to summarize data and extract meaningful patterns. Third, for each patient, a number of observations are made at each time point. Analyzing those observed features can be challenging, especially considering the existence of irrelevant features and the correlation between different features. To address those challenges and scale the analysis of clinic studies, many algorithms have been proposed to model patient status and understand disease progressions. However, most of those automatic approaches work as black box. They are hard to interpret and require careful hyperparameter fine-tuning to generate meaningful patterns. Therefore, instead of using an end-to-end model, in this study, we propose a two-phase method to provide users more control over the process of disease progression analysis. In the first phase, states are identified to describe different patient status using the value distribution of a set of observed features. For example, suppose three features A, B, C are recorded for each patient at each time point. Then we can describe different patient status using states based on the value distribution of the three features. After state identification, common state transition patterns are summarized in the second phase. Those transition patterns can help users better understand the different paths of disease progressions. Patients are further grouped based on the transition patterns to reveal potential correlations between disease progression and the patient-level features. Based on this two-phase analysis workflow, we propose ThreadStates, an interactive web-based visualization tool for state-based visual analysis of disease progress. This tool integrates existing sequence mining and clustering techniques with a set of visualizations to enable scalable and effective visual analysis. The interactive visual analysis provides users an interface to interpret and refine the results of automatic algorithms so that the users can better participate in the exploratory analysis and integrate the domain knowledge. ThreadStates consists of three views. The state identification view represents the identified space, and the other two views shows the overview and the details of the state transitions. I will walk you through all those three views and demonstrate the proposed visualization tool ThreadStates using a synthetic COVID-19 dataset. More information, design and implementation details, and other case studies are available in our paper. To start with, users can load the data to the ThreadStates. We support loading through the BioPortal API or uploading from local computers. Once the data is loaded, the visualization is updated and presents identified states and the state transitions. Here, color indicating different states. Those states are learned from time point features using unsupervised learning clustering algorithms. Here, users can select interesting time point features, remove unwanted features, modify the number of states based on the visualization results, and merge or split states as needed. Therefore, users are enabled to participate in the state identification process and integrate the domain knowledge, the observation about the data with automatic clustering algorithms to better collaborate and improve the state identification results. The state identification view consists of three visualization components. The scatter plot projects the high-dimensional time point features into a 2D space and offers user an overview of the state identification results. The glyph matrix explains the characteristics of each state by showing the value distribution of different time point features. Here, each row is a user-selected time point feature and each column is an identified state. The glyph represents the feature value distribution of the corresponding state. The aerial chart visualizes the temporal distribution of different states. The y-axis is the timeline and the axis indicating the occurrence frequency of the corresponding states at different time points. The three visualization components work together and enable users to examine and interact with identified states from different aspects. For example, in this dataset, the glyph matrix shows that the state A has a high occurrence frequency, which is indicated by the large width of the glyph, and the no feature values, which are indicated by the no glyph height. The aerial chart shows that state A commonly occurs at the beginning of the disease. In the scatter plot, state A, which is indicated by the green color, and state E, which is indicated by the orange color, are close to each other, indicating that they have similar feature value distributions. Therefore, we further examine state E in the glyph matrix. We can see here, state A and state E had similar value distributions for the first three time point features. The main difference between state A and state E is lactate dehydrogenase. At this feature, state E has much higher values than state A. The area chart shows that, unlike state A, state E commonly occurs in the middle late age of the disease. Since the feature selection will directly influence the state identification, we provide a feature manager to help users explore time point features. Users can select scores to rank different features. For example, here, we investigate features based on the rate of change and add several interested features. Interestingly, after adding those features, the points in the scatter plots are mixed together, indicating that the added features are not helpful for state identification. The glyph matrix explains the reasons of this scenario. At the rows of those features, glyphs are right angle triangles with the same height, indicating that this feature has the same event distribution on different states. Therefore, those features are removed. The identified states helps us effectively summarize different patient status, but how patients transit among these states? To answer this question, we provide a transition overview to show the patient state transitions. Those state transitions can be intertwining and need to visual cluster, making it difficult to identify insightful patterns. Therefore, we group patients based on the state transitions. For example, in this dataset, we can group those patients into two groups with distinctive transition patterns. This grouping significantly reduces the visual clutter and reveals clear transition patterns. Since the transition is directly influenced by the state identification results, we also support users to refine the state identification based on their observation of the state transition. For example, user may find that state A includes a large number of records and occurs both at the beginning and the end of the disease. Therefore, users can split state A into two states. The state transition is then updated. Meanwhile, to better understand the patterns in each patient groups, we'll present those patterns in an interactive table to facilitate the analysis and exploration. Users can rank those patterns based on their frequency and search interested patterns. More importantly, those patient groups may have distinctive patient-level features representing the correlation between state transitions and the patient features. For example, in this dataset, we can see that patient group 2 has less female patients and older ages. We can also see that patients in group 1 all survived while patients in group 2 didn't. This comparison helps us understand the correlation between patient features and the transition patterns of different states. Last, we can analyze more details about the state transition in the detailed view. For example, users may want to examine the medications the patient receives. To facilitate the analysis, users can also combine event features. For example, here, users combine event features that are related to the same disease. This helps users to identify the correlation between different event features and the relationships with states and state transitions. In conclusion, in this study, we propose interactive visual analysis of cohort start data through two phases: state identification and state transition summarization. We design and develop a web-based open-source visualization tool, ThreadStates, to support the two-phase interactive visual analysis. That's all. Thanks so much for your listening. You can find more information at our homepage and at our paper. You're also welcome to contact us to ask questions and share your feedback about this visualization tool.